This is a really exciting webinar. I talked a little bit about last time I was part of a group over in the Northwest border exercise where they were looking at an incidence of hogs, and part of the discussion was how do they handle animals out in the field that look sick and downtime to get them. Do they get them over to the lab? And one of the complaints from some of the wildlife and fisheries people was that the time to get something back to the lab and get results back was a week, even 2 weeks at times. And then you had animals out in the field and they didn't know what to do with them. Did they bury them? What happens if, crows or carrions get into the meat? Does it affect other populations? And there's no way they can control for scavengers unless they remove the animals. And then, there was some discussion, do we even want to touch the animals because we don't know what's in them? So I think our next presenter is doing Dr. Varma here at Purdue University is doing some super exciting stuff, well beyond anything that I understand, beyond kind of at a 50,000 foot level. But he's doing a lot of work in the professional public health space. We really call it a lot of point of care testing, and I don't know if that's the way they refer to it on the animal diagnostic side or not, but you guys did not come here to listen to me. We're going to let Dr. Merman talk about what he is doing, give a little bit of an introduction. If you have questions, feel free to type them in the chat, and then when we get to the end, we'll let Dr. Varma answer some of those questions if we've got some that ask, if that'll be okay. So without further ado, I'm going to get quiet and mute myself and unmute Dr. Merman.
All right. Good morning, everyone. Thank you for that introduction, Josh. Appreciate it. Josh, could you just give me a thumbs up? Everything looks good in terms of screen share and audio, video. Can you?
Yeah, you're good. You're good.
All right. All right. Thank you. So good morning, everyone. Welcome. My name is Mohit Verma. I'm an associate professor in agricultural and biological engineering. I also have an appointment in biomedical engineering. And today I'll talk to you about this problem of how do we get rapid results and how do we do this out in the field. And I'll take a One Health approach to this because animals is one application, but the type of technology that we'll see can apply all the way to humans, plants, and the environment as well. So happy to be here and looking forward to the discussions. So just a little bit of background about myself. I have a bachelor's in nanotechnology engineering from the University of Waterloo in Canada. After that, I went on and continued with my PhD in nanotechnology as well under Professor Frank Gu. And then moved to do a postdoc in Dr. George Whiteside's lab at Harvard for a couple of years. And then I've been at Purdue since 2018 as assistant professor and just recently as an associate professor. And while I've been kind of working on a variety of different things, one of the things has been trying to make lab-based tests much more accessible. So instead of having to send samples to the lab and wait for results, how do we make it so that we can get the results right there? That's really what we've been trying to develop. And we've been taking a One Health perspective. And what does that mean, really? So fairly recently, in the last couple of years, internationally, there's been this One Health high-level expert panel that has been established through, for example, the Food and Agriculture Organization, World Health Organization, Board of Animal Health, World Animal Health, and the Environmental Protection Organization. So they've come together to try to describe really what One Health means and how that relates to each other. So here's a framework that they've come up with to define One Health. And really what they're going after is that the health of animals, humans, and the environment is all interconnected. So you can see that on the left over here. And the actions of people affect not only the environment but their own health as well because of the interconnections, right? And the goal really is to look at it from a One Health perspective, and so that we can have healthy ecosystems, healthy animals, and healthy humans. So we're making more sustainable decisions, and we can also the societal decisions that we're making are at the different levels from a One Health perspective. And that requires, of course, communication, coordination, capacity building, and collaboration. So this is kind of like a big vision of where things might go. And the COVID-19 pandemic really kicked it off and showed that it is important to consider our ecosystems and animals as well, not just focus on human health, because things can come back and be a problem. So from this approach, a specific example of how things might work is shown for zoonotic spillover, which means like diseases that come from animals over to humans, right? We saw that with COVID-19. Right now, a lot of our work, as we saw with COVID-19, has been on the right side over here, which is more on the response side. So once we find out a disease, we kind of work on vaccines and increase healthcare capacity and so on, right? And then there's a lot of laboratory diagnostic that goes on as well. The goal of One Health, one of the goals of One Health is move towards more prevention. So move towards this kind of left side of this plot by taking actions to identify threats early and then reduce the chances of spillover, right? So there's a variety of activities that can be done. Some of that is fundamental science. So for example, just understanding how the dynamics of host and the potential pathogen play a role. Looking at what kind of human actions are responsible for potential spillover, those kinds of things. But where we play a role is kind of in the surveillance factor. So the integrated surveillance, as well as if the disease does outbreak, epidemiological investigation, we can build technologies that can do that faster rather than having to wait, so that the response times can be faster. And also we can reduce the cost because right now you really need a lot of expertise in personnel to be working in the lab to get it done. If we do it out in the field, we kind of bring that down as well. So we can potentially increase throughput, reduce cost, and reduce the time that it takes to get some of these results. So overall, really, if we do all that, the long-term vision, the impact from this One Health expert panel is just the world better being able to prevent, predict, and detect to health threats while improving all factors. So health of humans, animals, plants, and the environment, and trying to gear towards sustainable development, right? That's really kind of the blue sky, like ideal situation. Maybe in a decade or so we can get there, right? So that's really how they've been thinking about this. And what I am focusing on, of course, it takes a lot of people to come together to make this happen. So what I've been focusing on is just the surveillance and the detection part. How do we make that faster, and how do we make it simpler? So I'll show you a few case studies in animals, humans, and then the environment/plants to try to highlight some of the connections that we're already seeing. So animal health, when I got to Purdue, that's really where I started working on this problem. I was sitting next to a feedlot operator, cattle feedlot operator, and he was kind of mentioning that his cattle get sick. He doesn't necessarily know which antibiotics to use. If only there was a test that could tell him which pathogen is present, what kind of resistance might be present, then maybe he could make decisions better. So from that conversation, this project came out, and the overall idea is as follows. Right now, what they do with cattle is they look at healthy animals when they get sick. They're just looking at physical symptoms. So if they're off feed, ears are down, they're away from herd, then they might be sick. They bring them out, do a physical exam to check for temperature changes, changes in pulse, respiration, discharge, nasal discharge, and coughing. If those are not present, then they don't give them any antibiotics. But if it is present, it is classified as bovine respiratory disease. And then the current practice is kind of geared towards some antibiotic treatment, but almost by trial and error. It's basically, I say trial and error, but it's based on previous experience and also some clinical protocols. However, each case can be different, and we don't really have the data to guide the antibiotic treatment. So that's where we've come in. So we're building these field deployable biosensors where you can detect bacteria to know which bacteria might be causing the disease. We can detect resistance genes to know whether these bacteria are going to be resistant or not. And then we can also detect viruses. So if none of the bacterial one was found, if it was only virus, we don't need to use the antibiotics, right? So that's the idea, to make a more guided treatment and get all of these results in the farm rather than having to send samples off to the lab. Because you can do this in the lab, but the problem is it still takes too long, right? It takes several days. It's expensive. So people only use it if they're having a huge outbreak, and if cattle are dying, then they might send a sample to the lab. But by then they're not able to act on those results. So that's kind of the challenge. So to develop these types of field deployable biosensors, we have a workflow that looks as follows. In the case of bovine respiratory disease, you collect a nasal swab from the cattle, and you kind of resuspend this in water, put it on these devices or cartridges. We use paper-based cartridges where you can add the sample. Then these paper-based cartridges go into what's shown in a blue box, a heating imaging device that will conduct the assay and give you the result within an hour. Right now we can get it to about an hour. Some can be a little bit faster as well. And then you can read it either visually. So for example, similar to how you've seen COVID tests from the that we've used at home, you have kind of two lines. If the control should stay, in this case, we are showing it that it's yellow. And then the test, if it's red, it's negative. If it's yellow, it's positive. The key difference compared to a lot of the tests that are point of care or point of use right now, they detect proteins and antigens or antibodies, whereas we are actually detecting nucleic acids, which means DNA or RNA. What that allows us to do is potentially make it more sensitive, because there is an amplification step. So you can detect things that are at lower levels, lower concentrations. You can detect things earlier potentially. And then the second thing is that it is a colorimetric response, so it is visible to the naked eye if we want to make it visible. We can also get quantitative results by doing image analysis, and you will see some examples of that a little bit later. So how does this all work, really? It is based on a technique known as loop-mediated isothermal amplification. So you've probably heard of PCR during the COVID-19 testing. PCR was really the common way of doing things. LAMP is very similar to PCR or polymerase chain reaction. The main difference is that it can be done at a single temperature instead of having to oscillate temperatures. What that does is it allows you to make these devices portable, because then the equipment requirement is much lower. So within LAMP, so what we do is we put all our reagents on these paper-based devices, and within the test zone, what we will have is a target sequence. So this is what you're trying to detect. For example, in the case of bovine respiratory disease, this would be your bacterial targets, the sequence that is specific to that bacteria. We design primers, which are small sequences of nucleic acids, so DNA, which are shown in color over here. These primers will form loops and have an amplification reaction, which is really just the polymerization. So the DNA is becoming longer and longer. As that happens, as each molecule is added, actually, you produce a proton. This proton causes a change in the pH, and that pH change is detected as a color change. So that's really how this device works. And you'll see kind of the results from there. So early on, we with collaboration here at Purdue, with animal sciences, Dr. Tim Johnson, we measured the levels of bacteria between health and BRD, and we were seeing some differences in these levels. So the idea would be if you can detect the bacteria and estimate their levels, maybe you can predict which bacterium is causing the disease and therefore the treatment. So these are some of the pathogens that are responsible for bovine respiratory disease. So that's why we were looking at those as a starting point. And what we did then was design assays for those bacteria. We picked Histophilus somni, and Hymia hemolytica, and Pastorella multicida. So these are 3 bacterial pathogens. And then what you see over here are the different regions of the bacteria that we're targeting, LPB, LOLB, LOLA, and so on. And so those are just the names of the targets. And then on the x-axis over here are the different isolates. So PM would be Pastorella multicida, MH Mannheimia, and then HS Histophilus somni. So what you should see, and then the heat map is just the response. The higher the color, the more the response. So ideally, what you see is a diagonal line. So as shown in the red boxes, should show up positive. Everything outside should not show much signal. And we see that pretty much right on. So we can see that all the positives do react, and the negatives, there is some false amplification, but mostly they are okay. There was one isolate over here, which you can see that it wasn't really reacting, even though it should have to Histophilus. Well, we did some kind of troubleshooting there, and what we found out was it was probably mislabeled due to contamination. It wasn't actually Histophilus. So our test was kind of showing that too, that it's probably not Histophilus. So from this, we can design these assays, and we can show analytically, at least, we can get very high sensitivity, so 99%, and then specificity 89% about the bacterial pathogens. So far, these tests are still done in the lab. You use the fluorescence equipment just to do our initial screening. That's what we do, right? But to try to make it simpler and take it out in the field, we choose a subset of those targets, the assays, and develop a colorimetric assay first. So now we want the color to change so that it's easily visible. And we test that first in the lab to make sure that we're getting the right results, both for the individual bacteria, but also mixtures of bacteria. And once that looks pretty good, then we take it out in the farm and do the tests right there. So one of the key things over here, as I told you earlier, LAMP uses a single temperature, so you just need a heat source to do these tests. What we did was, instead of using something fancy equipment, just because we wanted to do it quickly, easily in the farm, we used a sous vide cooker. So you see over here, it's a consumer grade sous vide cooker that is used for cooking meat. We set it to 65 degrees Celsius or 149 degrees Fahrenheit, and then we put our tests in there. And then when you have a positive result, the test will change to yellow. If it's negative, it will remain red in this case. So we took this, we kind of took this and developed the colorimetric assay. So here is just an image of what the colorimetric assay might look like. These are just the different bacterial targets, and then the bottom is negative, the top is positive. So you see a change from red to yellow when you have a positive response. Then we can actually go out in the feedlot and test these. So you see in the first step over here, one of the students is collecting the nasal swab from the cattle. This swab is then just resuspended in water. So that's what this other student is doing. We take some of the water and then put it onto our assay tubes. It's a little bit hard to see over here, but that's what that one is. And then we place it in the water bath, the sous vide cooker, and observe it from the outside to look for the color change. And we saw pretty good results, so about 60 to 100% concordance between what we could get on farm and in lab results. So the 100%, and the variation was really dependent on which types of targets we were looking at. The low one was we thought so one of the primer sets was reacting to the complex samples, which we didn't expect to, because so we are redesigning that to try to bring it closer to 90 to 100% concordance. So really, but what it's showing is this proof of concept that yes, you can get the results within an hour, and you can get the results almost just as good as what the lab results would be, right? So you can make the decisions right there. So with that, now we can start to think about the interconnections between different industries, right? So we looked at the cattle feedlot, and what I'll kind of jump into a little bit is environmental health, or you can see what gets deposited on plants a little bit, because this is a food safety issue. So, for example, we looked at what happens within a feedlot from an animal health perspective, right? But oftentimes, what is now happening is these feedlots might be surrounded by farms which are growing fresh produce that humans will consume. Now, these fresh produce are often not processed, right? So we use them, we just kind of wash them and eat them. So they have to be very careful about what they're being exposed to. So one of the hazards in this kind of setting is that the fecal matter, for example, from the feedlots might get aerosolized and deposited on these fresh produce, and then that becomes a food safety issue, right? Um, and this is happening more and more because we have an increased demand for food. We have limited land, so people need to grow things closer and closer together, right? Both for meat as well as for plant-based growth. So that's why this is becoming a challenge. And where we come in is try to assess the risk that these farms have from nearby operations. And we do that by measuring how these aerosols are being deposited, and also trying to make it specific, because right now what they do is they have distance guidelines. So depending on the size of operations, they'll tell you, okay, you need to be this many meters or this many feet apart away from the feedlot. However, that is not very specific to the site. So in that case, it may be that you're just too far and you're not using the land in between, even though you could have, or you're not far enough because under certain weather conditions, it can still be unsafe. So we're trying to give it some more specificity, give a little bit more science and data behind it. So to do that, we take the same platform that I described earlier, trying to detect DNA, RNA on these paper-based devices with colorimetric response, but we build a little bit more to it for this application. So what we do is we design these simple collection flags, we call them, which is just a kind of bamboo stick over here, bamboo skewer over here, and a piece of plastic that's attached to it. Because we were looking at initially leafy greens, so that's why we chose this design. It's about the same height as lettuce and so on, and we can place these skewers all around the field, and then get a distance-dependent response of what might be depositing, what kind of hazards might be present. So we test them around animal operations just to see, does this idea work, and what kind of levels can we expect from if you were growing it right next to animal operations, which usually doesn't happen. There's some distance, but just to see a positive control, we do that. And then we test it out in the field to see what are the levels if you have very low risk, right? What is that range? And to enable this, what do we detect, right? So I think anytime people think of food safety, they might think of, okay, you can just detect foodborne pathogens, but that is actually quite challenging for two reasons. One, the foodborne pathogen levels are generally, if there are, they'll be very, very low. So it becomes really difficult to detect them. And that's a challenge even with the current testing methods. Two, anytime you mention pathogens, the industry becomes very sensitive, and rightly so, because there's so many reporting requirements and regulatory requirements that we won't really be able to do these field tests very easily. So instead, what we do is use an indicator bacteria for feces. We use Bacteroideals as our indicator, which is present in the mammalian gut in very high concentration. So about 30 to 40% would be covered, which allows us to get pretty high concentrations, about 10 to the 9 to 10 to the 11 copies per gram of feces. So if you get even a little bit off being distributed, we can probably detect it. It is obligate anaerobe, which means that it won't really grow outside of the mammalian gut in an aerobic environment, which means you can maintain similar levels. So it helps make this a quantitative indicator because you can look at the levels and say, okay, yes, this much physical matter might actually be present here. Naturally, it has low abundance unless it's coming from fecal matter. From non-fecal sources, the abundance is pretty low, so that gives us a little bit of specificity. And then you can also get host specificity if you need to. You can determine which host it was coming from. Was it coming from humans, for example, or cattle, or swine, or poultry? So we can do that kind of analysis as well. So we developed an assay to detect bacteroidals, and then we used our collection flags that I described, placed them around these different animal units, and tested the levels, right? One of the things that we're doing is comparing the response in lettuce. So that's kind of what you see in the middle versus our collection flags. Because lettuce was just grown in greenhouse as a control. And what we're seeing is that the responses are much less reliable in lettuce because of the surface characteristics. When you go to swab them, they don't give you very good outputs, whereas the collection flags give you very clear, more repeatable outcomes and clear trends. So that's why we kind of stuck with collection flags. And what the other thing we noticed was, depending on the type of operation, we have different levels. So the swine unit, which you can see is pretty close distance and pretty high density, we have pretty high concentrations, up to 10 to the 4 copies per centimeter square. And then in the cattle unit, you still see some distance dependence, but the levels are lower compared to the swine unit, right? So this was promising, just to show us the range of bacteroidals that we might be able to see from this method. Then we went on to see a field that is away from our operations, normally would be used to grow leafy greens and see what happens. So the schematic is kind of shown on the left. And what you see in plots B and C, which are just in lettuce and the collection flags, is that the levels are extremely low. So in the previous plot, this axis went up to 3, and it's a log axis, right? So it's 10 to the 3 copies per centimeter square. Whereas here, we had to bring it down to 1 just to see even some of the very small levels. So these levels, and in most cases, most of these samples, which are actually shown in kind of this light yellow color, those are actually all data points. They're just 0. So we don't see anything, which means the levels are extremely low, which is a good confirmation of confirming that from non-fecal sources, the level can be very low. So now we have a baseline of 0 to 2 copies per centimeter, all the way up to 10 to the 4 copies per centimeter. And we can think about determining risk in between those levels, right? So for example, the idea would be to test the samples right there using some of our techniques that can be done out in the field and get the results right there. So for example, some of the things that we did was plant these collection flags, collect them in the field. So this was in animal operations. The one on the right is in the leafy greens operations. In that case, we're doing it in the back of a van and doing the test right there. And then comparing the results that we get from our device, which we're calling a decode, to qPCR when we bring it back to the lab. And again, these results you can get within an hour-ish. And then whereas this one, you'll have to ship and then do the test and then report the results. So we're seeing very similar results. And in terms of the limit of detection and what you would expect, the results were pretty much in agreement. So we're getting about 85 to 100% agreement with the lab-based results. So this is really promising now because, again, you can do this much faster. You can do it in the back of a truck and you can do it out in the field. So that becomes useful. And the overall idea then would be that you place these flags, collect the samples. In this case, we do leave them out for a week. That period could be smaller or shorter or longer, depending on the application and the environment. We do the assay preparation right there in the field, collect the sample, add it to our devices, and then we get a response. A negative response would look basically red on both pads, and then the positive would look, for example, yellow on the reaction pad. And then we can build these heat maps of where the risk might be, whether it's from small point sources or from more controlled neighboring land use types of issues. Based on this risk map, then you can introduce interventions. For example, You can have vegetative barriers or buffer zones that will prevent those aerosols from coming in and protect your produce, or you can grow things that are not meant for direct human consumption, could be used for something else. So those kinds of decisions can be made based on these data that are collected out in the field. So as I mentioned, now we move on to, because we eat these things, right? So that was a food safety issue. And we can now start thinking about human health. And the example that I'll present here is with zoonotic diseases, specifically COVID-19. And the idea is very similar to what you saw for bovine respiratory disease. Now we're thinking about collecting human sample. So in this case, we designed the assay for saliva. It could be for nasal swab as well, and other sample types, but we'll look at saliva now. And this other process is basically the same, added to the paper pads and then read the result. So initially, we do the same thing where we need to design an assay for SARS-CoV-2 in this case, right? And now we can detect RNA by adding another enzyme that allows the reverse transcription from RNA to DNA and then detect that. And then you see a similar colorimetric response where negatives stay red, pink, and then in this case, positives will change to yellow. In this case, we're seeing 30 minutes, between 30 to 60 minutes is when we get a response. We take these assays and, as I mentioned, we want to make this simpler so that the user doesn't really have to handle too many reagents and so on. So we want to put this on paper-based devices, and we do that over here. So what you see is some troubleshooting and optimization to get the assay to work on paper. One of the challenges early on we found was if we just put all the reagents on paper, they turn to yellow upon drying just on their own, even if you don't have any target. So over here, the negative one should be your control and the positive one has your template. So the negatives should stay, actually, in this case, we're not doing any amplification. So we're just trying to see, they should both actually stay red instead of turning yellow so that you can actually use them for result interpretation. And what we did was we eliminated each of the ingredient one at a time to find out which ingredient was causing this color change. And we found out that it was the ammonium sulfate that was the issue. So to make it work on paper, we got rid of the ammonium sulfate and found a replacement for it so that we can actually get the assay working on paper. And then the workflow looks as follows. You collect your saliva samples. We use this device, PureCell, which makes it simpler to collect saliva instead of drool tubes, for example. And then re-suspend that in water. Then add the sample to your pads, so the control and the SARS-CoV-2 zones, and then incubate it. In this case, it could be an incubator, it could be a water bath, as you saw. It could be other heater sources, simpler heater sources as well. And we got the results that you see over here. So in this case, these were saliva spiked with inactivated SARS-CoV-2 because, of course, getting the active testing was challenging. So what we did was, first of all, in panel B, you see kind of a limited detection study. Here we have different copies per microliter of saliva of the hidden activated virus. And you can see that the limit of detection is about 200 copies per microliter. So the 3 replicates where you see a change to yellow, orange, those would be considered positive. The ones where you don't see or not reliable change, then we consider them negative or is not very reliable at low limits. So that's reasonable for this kind of assay because it is in a complex medium. If you extract the DNA, RNA, you can make it even better. You can go down to 50, 25 copies, or maybe even lower. And the negative remains red or pink, and that's a clear color difference. Then we put these pads onto these kind of strips. So what you see over here is the 2 pads for the different targets. We have some polystyrene spacers to prevent cross-contamination, double-sided adhesive to make it all stick, and then MLNX backing to support everything. That makes a nice thin strip that can be used for adding the samples. And we see a typical response where the negative stays red and then the positive reaction zone turns to yellow. You can compare that visually to this kind of color scale where we set a threshold of what should be considered negative and positive. And when we do that, what we see is that there is a pretty high invalid rate. So people report results as invalid a lot of times because we're getting some kind of changes on the outside, not uniformly, and people are reporting that as an invalid result, just when looking at the strips. But if we discard that, then the sensitivity is about 76%, accuracy about 90%. And to build all this, the cost of materials is around $10 per test. A lot of it comes from the sample collection, so it could be brought down, but that's kind of the order of magnitude. What we did then was to improve these results. I mentioned image analysis. So we can take the pads and look at the RGB intensity, in this case, the green color, and see what response we get from the negative samples versus the positive samples. And then we actually get a much better response because there's a clear distinction. These are at different concentrations between the negatives and positives. And you can see this ROC curve, so the receiver operator characteristic curve, shows a pretty, it's a really good response. So a perfect response would be basically a square curve over here. So it's pretty close. And then a coin flip would be this diagonal line. So the blue line shows where our test is, which is pretty good. And then you can increase the sensitivity and specificity to 97 and 100% pretty much for an accuracy of 98%, because now you don't get any invalid results. We can get all of them to be valid. So that basically what that tells us is for these tests, it's probably better to include image analysis into that heating device. And that's what we're doing right now. To make it all kind of work together and easy to use, we worked with a couple industrial partners during this COVID project, where we made an easy to use cartridge, which you see in here with the green component. What is inside these cartridges, these two pads, that's what we're seeing as the color. That's where all the reagents are. That's where you're seeing the color change. And then they also built a heater, which is simple to use, so that you can just kind of press a button and it turns on and does its heating cycle. And then on the right is the collection kit, and this is where the cartridge would be. So you can also compare it to the color chart visually if you wanted to. So it just shows you what the package might look like. The size over here, this is about the size of a cell phone, a little bit thicker. So it's pretty portable, pretty small, pretty easy to use. And that's kind of the vision, right? To make these things portable, easily deployable, hopefully out in the field, and get the results much more rapidly. So you saw a few vignettes of different applications. Now we're taking the SARS-CoV-2 test and bringing it a little bit further. So we have a recent project where we are trying to make it a universal test, right? So in the sense that not only test humans or specific animals, but all animals. So can we test all wildlife, livestock, companion animals using this kind of one test and get a result? Because then that helps you with surveillance. If you have reservoirs for SARS-CoV-2, you can do that surveillance much better, and we can use it across different stakeholders. So that's what we're working on now, and that's kind of really starting to take the One Health perspective on these emerging diseases. You can also expand it for human diseases, look at influenza as a target, for example, which we're working on as well. For bovine respiratory disease, we saw the example from bacteria, but we're also looking at antibiotic resistant genes and viruses to help the overall decision making. We have a project on African swine fever, which is the biggest animal pandemic, really, which is a problem. And you basically, luckily, it's not in the US yet, but we need to be prepared in case it gets here to get a rapid response. So that's what we're building. We have an assay for avian influenza. You saw the big issues with avian influenza that happened earlier. So just getting faster response would be the goal. You already heard about the food contamination. So we can do the fecal contamination detection out in the field. We can also do detection of pathogenic bacteria for specific samples when it's relevant as well. And then finally, we also look at plant diseases and traits, which can be, again, used for faster decision making out in the field. So overall, really, the vision is we've looked at these systems almost individually. So you saw applications of animal health, human health, a little bit of environmental from a food safety perspective, but all of these are interconnected, right? So how do we use these tools to determine how decisions in one affect the other? That's really where we're going, trying to build this integrated One Health approach. So of course, the work takes place thanks to my lab members. And usually I highlight which members have worked on this project, but in this case, it's almost everyone. So I didn't do any bolds. Pretty much everyone has worked on it because we're doing a One Health integrated perspective. And that ranges all the way from postdocs to grad students, undergrads, research assistant, as well as visiting scholars. And they're really the ones who do all the work. And we've gotten funding from a variety of sources to support these projects, and those are listed over here. Finally, an announcement, which before I do that, I do have a startup company which is working on commercializing these technologies. So they're taking it, they're licensing it from the lab to translate it out in the field to turn it into products so that people can use it much more easily. So that's my conflict of interest here. The work was not funded by Kershey, at least the one that was presented here. Finally, an announcement and from a colleague, Dr. Gray, who I believe is in the participants. There is a One Health symposium coming up, organized by UTMB, and you're welcome to visit this to register. Seems exciting. So thank you all, and I'm happy to take questions at this point.
Yeah, so please feel free if you want to type in a chat box. I think Marcy can take people off mute if you have questions. Thank you so much for just the presentation. That's really interesting. And as somebody who's worked in the healthcare field before and did, we had in the public health and human health, we had the LDTs for COVID-19 testing we were developing and respiratory testing as well. Do you see, just out of curiosity, is the biggest hurdle to make it friendly for not just farmers, but wildlife and fish and game agents when they stumble upon, the heating element and all that? Is that the biggest kind of field readiness hurdle you've got to overcome?
Yeah, in this case, depending on the application, we can actually turn it into an integrated device. So most of the applications that we've looked at has been where you want to test multiple samples quickly. So that's why we have a dedicated heater, and then you kind of have these disposable cartridges, right? If you have areas where you're not testing as frequently, but need the results pretty quickly, we've been thinking about just an integrated heater into the disposable components. The whole thing then becomes disposable, and that might be more applicable where you have a few cases, not as frequent, which might be the case for wildlife. So that is a possibility. We haven't developed that, but if we get the right applications, I think we can do it.
Out of curiosity, this is a question. If others have, feel free to chat away. I'm assuming the nose swabs work much better on animals as opposed to humans, just because it's easier to put a swab up an animal's nose. Would this continue to work on an animal? Back to the hog situation. If you found a dead hog out in the field,
yeah,
is it viable to use it on a hog that's dead or an animal you find dead out in the field?
Yeah, so the last project that I mentioned that we're working on currently, with looking at expanding SARS-CoV-2 tests to other animals. In that case, that's where we're testing what happens if you have dead animals. So for example, deer, right? A lot of deer have been carriers of SARS-CoV-2, and we get some samples from hunting or other sources, and we're testing that right now. So we're testing how well the swabs work in the dead animals versus not. I think the key component is whether the virus or whatever you're testing is still there or not. So if the animal's been dead for a long time, then the target might not be there. So that can be a challenge. But if it's fresh enough, then we think the test should still work just as well.
Yeah, and speaking of, I'm just going to ask a question here because I've got a couple. Speaking of wildlife, this was actually brought up in that session, even though it was about hogs. But what about CWD obviously is a big one, and then EHD, the hemorrhagic disease. Is that something you guys are looking at as well?
Yeah, so CWD, we not yet, and it's a little bit challenging because I believe it's caused by prions, if I'm remembering correctly, rather
That's
than
correct.
nucleic acid. So the tests are all designed towards nucleic acid detection, so we can't really detect proteins, for example.
Okay.
So that's a challenge there. The other one, I'm not sure what the target is. If it's nucleic acids, we can do it. If not, it becomes a little bit challenging.
Yeah, I don't know right off the top of my head, but I know that affects a lot of deer
Mhm.
population.
That's
In
the same question we got as well when we were kind of looking for samples for SARS-CoV-2. They're like, "Oh, can you do it for this as well?" We're like, "Well, it depends on the target." So, if it's nucleic acid, so we can do a lot, but not if it's like proteins, at least not this way. But
There you
it's
go. Somebody just posted there. EHDV is a real virus.
right. So if it's a virus, we can do it. We feel pretty good about it.
Not an animal scientist or I'm an economist by trade. So so me understanding all the implications, but I thought the with respect to the plant testing, is that something that's you think will be looked at? And I know we have some plant people on here looked at in the future, where you look at, hey, what's the what let me go with all of this ESG work that's being done about supply chain management, right? Yesterday, the dairy group agreed to participate, several of the companies in dairy, for methane production, but, they're looking at sustainable supply chains and making sure you're doing everything to continue to be in the supply chain, and then ultimately receiving funding if you want to expand operations. Do you see that as something coming down the road, that if I have an animal operation and then there's a commercial food production operation, that I'm going to have to start doing some of that testing before, certifying my plants before they go off to production?
Yeah, that's actually a very good question. We have not thought about it that way because we've been looking at just the food safety perspective, because we've come from almost the project was triggered from the produce side of things. But and I think the challenge has been that there's different governing agencies for the animal production and then the fresh produce production. If we can get those agencies to talk, I think that, and I think we should, instead of being like adversaries, then that makes a lot more sense to try to say, okay, for this environment, this is what your impact is on the neighboring land use, and this is what we can do. So I think it could be used there. We haven't been approached in that way, but I think so. Yeah.
Yeah, I think that's coming in terms of, everything I've been reading about the supply chain and what they're trying to do to make sure you can still stay part of the supply chain, and being able to quickly, recognize if food enters the system, right? That we don't have to shut down. We don't have to call back every single truck we just sent out. We have to
Exactly.
call back
Yeah.
this single truck because
Yeah.
that was the affected production
So
area.
yeah, the traceability part for sure, right? Because right now they just do, yeah, this product and everything comes back. If we get much better at that, tracing where it came from and which specific was the cause, I think that's where we can help with some of these technologies to do that tracing faster.
I have one more quick question, then I'm asking because I have, but I don't see any popping up in the chat there. Do you think this has applications to ports of entry for when we're coming in for ports where we can test items?
Yeah. So, for example, African swine fever, that's one of the places where it could be tested. It could be used, right? Because if it gets introduced through meat or something, that can be a big challenge, big problem. So for sure, it can be applied there. Same thing for plant diseases as well. Some of these diseases can be high impact or high consequence. If we're getting things coming in, if we need a rapid result, instead of kind of holding and sending to the lab, we can do the testing right there. So those are definitely two applications. Yeah.
We have a question from, if I mispronounce it, I'm sorry, from Sanjeev.
Mohit, this is Sanjeev. Hey, excellent
Sanjeev.
job. I really appreciated your presentation and talk about the LAMP system per se. My question is more as a pathologist, as a veterinary pathologist. So what happens is, many times Mannheimia, the list of organisms that you had mentioned, Histophilus, Mannheimia, Pasteurella, these are normal inhabitants of the nasal tract, right?
Yep.
So it's sometimes when I submit a sample, if it doesn't come from the lungs, affected lung, I don't even, I mention it, but I don't call that as the cause of the disease, right? And so that's one, and it's on the same vein. You talked about bacteroides. Bacteroides, even though it's an anaerobe and it doesn't form, spores, but these live in, especially if you're looking at the DNA, you can have those in the soil for months. We have done studies where it's like 13 months they're still in the soil, right? And cold, heat doesn't matter, And so what I wonder is, have you thought of not only the organisms coming from the fecal contamination that comes from the farm, but it's in the soil,
Right.
right? It's one of those things where you have really high sensitivity. It can really hurt you because, what about leftover, it got dusty, right? It just got dusty, and now that organism is sticking to this filter.
Yep.
So I don't know if you have thought about those things, especially as in our field in veterinary medicine, even organisms that we isolate, people don't give a lot of value to it because you can isolate mycoplasma from every lung sample, affected or unaffected, especially those affected, and people would still say it's just a contaminant, So just curious about your thoughts on that.
Yeah, no, actually, those are great questions. And as we've done more and more research, what we realized is one of the things is generally the baselines aren't very well done, right? So, for example, like what are the levels of these commensal bacteria in cattle in nasal swabs? That wasn't done until it was done preliminarily, but not extensively until we started doing it. So we actually have done, Dr. Johnson has done this a lot. We've collected nationally a lot of samples and looked at the levels of these bacteria. And when we do that sampling, what we see is there's regional differences. So in some cases, the result that I showed would be useful because you can see levels going higher or lower. In other cases, it may not be because they are actually just present commensally. So then what we're thinking about right now is two potential routes. One is you can start to look for more specific targeted pathogenicity genes rather than just looking at the species level, which might be more useful. The other potential use is look at the antibiotic resistance genes, because then maybe regardless of which pathogen is present or not, it could still be used to guide which antibiotics are more likely to work. So that's kind of one way of doing that. The other one is the same problem where we don't have baselines in terms of what are the levels of bacterial all over the place in these different conditions. And so if you use a binary test, then you're right, like the sensitivity hurts us, and if it's just a yes, no, it's a challenge. And that's why we're not, in that case, doing a binary test. Actually, in most cases, we're moving away from binary tests and looking at the actual levels, the concentrations that we're getting. And we think even with a portable device, we can get some quantitative levels. So we can say, okay, it's 10 to the 2, 10 to the 3, 10 to the 4, and so on. Then we can use it to see, okay, actually, right next to the feedlot, it's 10 to the 4. Right here, it's 10 to the 3. And now it's dropped 10 to the 2. And we think that 10 to the 2 is comparable to what baseline levels should look like. So these should be safe enough, and then these where we should not harvest, or we should build additional interventions. That's how I envision this being used. And rather than your typical pathogen testing of yes, no, it's a problem, actually look at the map and see where those issues are. And it does actually, in addition to technology development, and we have to do this, it does involve a lot of these baseline measurements, which are not very well established. So we are actually starting to publish some of that work as well, just showing, okay, like for example, the plot that I showed in the soil, at least in the leafy greens, it is actually really low. That's why it's not getting onto the leafy greens, even if we leave it out there for a week. In some cases, it might be that it's there for a while, but then we can do those tests and find out, okay, this is the baseline level for this field or this location is a little bit higher. If it has not caused disease in a long time, then maybe that should be your threshold. The threshold would be higher. So it becomes more customized to the site. I think that's how I envision this being used going forward.
Well, thank you. And the talk about, you mentioned about some of the virulence genes. I think you have LKTA there just for Mannheimia. Your target is leukotoxin A,
Right.
which is present in most organisms. I think there is this phenomenon of a viral infection that really has to precondition the lung for the bacteria to invade. Right.
Yep.
So just presence. And again, I'm very interested in learning how the bacterial load in a normal animal versus not, in cold temperature versus not, in a highly, high population or density condition versus not. Those things would be pretty interesting. And going back to, so I think the idea of using virulence factors is good. It targets, but again, most organisms, even those that live there as a commensal, carry these, and I have never, in my experience, and other people, I can't see who else is joining. Other people who are microbiologists probably can help too, but it really doesn't. I have never isolated a Mannheimia that doesn't have LKDA.
Right. Okay.
So,
Sure.
So that's good. Thank you. I appreciate it.
Yeah. I mean, it's an. I'm not saying the problem is solved yet. I think we've built the tools now to try to tackle the problem a little bit deeper. Is what we've learned. We have more questions after doing this project than the answers. So yeah, your points are right on.
Yep. Give just a couple more seconds to see if anybody else has any more questions, even in the chat. If not, while they're doing that, Doc, thanks for the wonderful presentation. I think what you're doing is going to be, kind of a key to frontline prevention of a lot of the spread of these diseases, to make sure we can kind of catch them very quickly and not have, outbreaks, whether it's having some of these at, weigh stations on interstates where we're checking random checks of some, livestock just to make sure, or, if we've got hot spots coming up, or at ports, I think are huge issues. And then I personally think, until I saw this, I did not know about the plant part, that it will be a key avenue for ESG as farmers, as we're trying to make sure we can get funding and be certified ESG compliant in, these big, huge multi-conglomerate supply chains. So thank you so much. I want to tell you, I am jealous of that beard. It's grown a lot since the one picture you had. I have to let mine grow a little bit more to get there, but I like it. So thank you so much. We appreciate everybody for attending. Doc, again, enjoyed it. I hope everyone has a wonderful day, and join us next month when we have our next seminar.
All right. Thank you, Josh, for having me. And thank you everyone for your questions and attending. Thanks.